Good morning, viewers. Today, I want to tackle some basics about genetics, which is the first topic in Form 4 syllabus. Now, with me here, I have a question that reads, in an experiment, a variety of garden peas having a smooth seed coat was crossed with a variety with a wrinkled seed coat. All the seeds obtained in the F1 had smooth seed coat. The F1 generation was selfed, and the total number of F2 generation was 7,324. Now, to our first question, part A, using appropriate letter symbol, work out the genotype of the F1 generation. So if you come across such a question, number one, the question itself will give you a clue on how to deal with the genetic cross when you are crossing the various characteristics. So what you need to note, number one, you have been told that there are two varieties of plants here. We have a variety of garden peas with smooth seed coat, and we also have a variety with wrinkled seed coat. Now, the clue here is that you are told all the seeds obtained in F1 had smooth seed coat. What does that tell us? Number one, this shows that the smooth seed coat characteristic is dominant over the wrinkled seed coat. Now we are going to deal with a case of complete dominance in this question. Now, when we have complete dominance, we will have the smooth seed coat. Smooth seed coat being dominant will have letter R, capital. Why do we use capital R? Because it is dominant gene. Then we have wrinkled seed coat. So the wrinkled seed coat will have a recessive gene, which will be represented by small r. So the small r represent recessive gene, while capital R represent dominant gene. So smooth seed coat is dominant over the wrinkled seed coat. So when you want to have the genotypes of these characteristics, we are going to have the smooth seed coat having R, and then wrinkled seed coats having small r, because it is recessive to that one of smooth seed coat. Now, we have been told from the first question that using appropriate letter symbols, work out the genotype of the first filial generation. So F1, if you see F1 refers to the first filial generation. First filial generation refers to the genotypes of the offspring from the first crossing. So here, being that we have our genes, we have smooth seed coat and we have wrinkled seed coat. Therefore, when you want to carry out a genetic cross, number one, in a genetic cross, you must identify the phenotypes, you must identify the genotypes, and then you must use correct crossing lines, and also you must identify the parents. So here, we are going to have our parents. We have the parental phenotypes. We will start with parental phenotypes. We are going to have the male and also female. Then the phenotypes will have smooth seed coat and then wrinkled seed coat. Then, after phenotypes, we are going to look at the genotypes, the various genotypes of these parents. So, the genotypes, we had already identified the genotypes of the smooth seed coat, being that it was dominant over the wrinkled seed coat. So we are going to have R, and then this one will have small r because it is recessive. Then you must have what we call a cross between the genotypes. We are not going to cross the phenotypes, but we are crossing the genotypes. So after that, then you go to the gametes. When you go to the gametes, these gametes will be derived from the genotypes. So we'll have this, and you circle it using a complete circle. That one again. The other one also you circle using a complete circle, like that. Then after getting your gametes, we are now going to look at the fertilization, or you can call it fusion, when now the various gametes are going to fuse with one another so that we get the F1 genotypes. Now, when you want to use the fusion lines, make sure that the lines are complete. Number two, we can also use what we call a Punnett square. So the Punnett square also will help you in getting the various gametes. So in this case, we are going to look at this question, and we are going to use a Punnett square for us to obtain the F1 generation. So we are going to draw the Punnett square. Now, from here, we are going to divide it. Then we are going to have this, like that, and then divide that diagonally like that. So we are going to have this one, and then we'll have the female there. So all the gametes for the male will be placed at that point, where we are going to have the gametes of the smooth seed coat. Then here the gametes for the wrinkled seed coat. So after drawing that and indicating your gametes, we are going to start our fusion, where we have this gamete fusing with this one to give us this. Then this other gamete will also fuse with this to give us that. This one with this will give us that. And lastly, we'll have this fusing with this other one to give us the last one. Now, so when we look at this, these are what we call F1 generation. Why do we call them F1 generation? I earlier explained that F1 generation refers to the first filial generation, meaning the gametes of the offsprings that have been obtained from the first parents. So when we were asked to use appropriate letter symbols to work out the genotype of the first filial generation, we have identified the phenotypes of the parents, we have identified the gametes, and now we have crossed and we have obtained the genotypes of F1 generation. So when you are asked about F1 generation, you will say that F1 generation, all F1 generation are heterozygous. Why do we say heterozygous? Because we have capital R and small r. When we have capital R and small r, these represent heterozygous states. When we have capital R and capital R, this one is now homozygous state. So we can have homozygous state, recessive or homozygous state when we have both the genes having dominant state. So the F1 generation or the F1 genotypes are the four. We have four genotypes here that represent the offsprings of the first parent. So we have used the appropriate letters and we have worked out the genotypes of the F1 generation. So let's look at the second question that will help us to also obtain the next solution. On to the second question. From the information above, work out the following for F2 generation. The first one is genotypic ratio, then we have phenotypic ratio, and lastly we have the wrinkled number. So when we are told from the information above, it means we are going back to the first genetic cross that we did that involved F1 generation. So if you have been told from the information above, work out the following for F2 generation. And from the main question, you were told that the F1 generation was selved. And the total number of F2 generation was 7,324. That means whenever you self F1 generation, you will obtain F2 generation. So we are going to self F1 generation. From our first cross, we obtain all the four gametes or the four offsprings of F1 generation, and we realize that all the four gametes were in heterozygous state, meaning all of them were smooth seeds. Why do we say smooth seeds? Because we had capital R and small r, heterozygous state. That means capital R, which represented smooth seeds, was dominant over the small r that represented the wrinkled seed. So here, for us to obtain F2 generation, we are going to self F1 generation. That means we are going to start with the parents. We are going to follow the same steps that we had in F1 generation. We have that. Then we go to the parental phenotypes. We are going to have all were smooth seed coat. This was also smooth seed coat. Then we go to the genotypes. So we have phenotypes, smooth seed coat and smooth seed coat. Then we go to the genotypes. We are going to have capital R and small r. Then we have the cross, capital R and small r. And then we go to the gametes. We'll have this complete circle, this other one, then we go to the fusion or even fertilization. So here we are going to have our Punnett square. We'll have the Punnett square like that. Then here we have the female. So the gametes for the smooth seeds, we have this and this other one, the same to this side. Then we start our fusion. Capital R with capital R will give us that in homozygous state. Then we'll have this small r and capital R will give us capital R and small r heterozygous state. The same to this one, another heterozygous state, and then we have homozygous recessive. So this one is our F2 generation. So these are the F2 generation. These are the gametes for the F2 generation when we self F1. So remember, we had obtained F1 generation and now we are selfing F1 generation to obtain F2 generation. So we were told to use the information to obtain the genotypic ratio. When you are asked about genotypic ratio is always given in form of genotypes. Why do we say that? We don't express genotypes in words. Instead, we express genotypes using the various genotypes that have been obtained from the crossing. So the genotypic ratio here, we are going to have, our genotypic ratio will be, we are going to have this one homozygous smooth seed coat. Then we have here two heterozygous. We'll have two heterozygous, and then we'll have one recessive. So this one represents homozygous smooth seed coat. This one is heterozygous, but still smooth seed coat because capital R is dominant over small r, and then this one represents wrinkled seed coat. So when you are asked about genotypic ratio, this will be our genotypic ratio. Then we are also asked about phenotypic ratio. Phenotypic ratio is always expressed in words, and whenever we have complete dominance, the homozygous state will combine with the heterozygous state, so that we have these two will give us smooth seed coat, while this one will give us the wrinkled seed coat. So when we look at this case, when we want to get the phenotypic ratio here, phenotypic ratio will be, we'll have 3 smooth seed coat to 1 wrinkled seed coat. That is our phenotypic ratio. So I've said genotypic ratio will be expressed in form of gametes, not gametes but genotypes, while phenotypic ratio we express in words. We have 3 smooth seed coat and 1 wrinkled seed coat. Why are we saying 3? Because 1 homozygous smooth plus 2 heterozygous smooth seed coats, that will give us 3, and then we'll have 1 wrinkled seed coat. And therefore, lastly, we have been asked the number of the wrinkled seed coats, being that we were told the total number of F2 generation were 7,324. So to obtain the number of wrinkled seed coats, this is the total number of the seed coats plus the smooth ones. But now we want to obtain the number of only the wrinkled seed codes. So what we do here, because we know that for any gene or for any gamete to be transmitted or gene to be transmitted, the probability will be a half. So we'll have this, a half, a half, a half, and a half. So for any gene to be transmitted, the probability is a half. Therefore, when we want to obtain the number of wrinkled seeds, we come here. We look at the probability of this gene R being transmitted from the parent to offspring. So we are going to have this half times this half, which will give us a half times a half will give us a quarter. How do we obtain that a quarter? I've said a half and a half. When you multiply them, you are getting a quarter. So this one will have a quarter. You multiply by the total number, which is 7,324. So the value you are going to obtain from here, you will obtain a value that will represent the number of wrinkled seed codes that had the recessive gene. So a quarter, when you multiply here, you will get 18. And then you will have 3 and 1. So we'll have 1831 as the number of wrinkled seed codes. Now, that one is an example of how we carry out a genetic cross, and we'll do more examples as we move forward. Thank you.